Today, my guest is Dr. Nabora from the University of California, specializing in cell, cell senescence, long aging, and regeneration. 2 weeks ago, she has just published a paper related to senescent cells, the science. And she found out that the senescent cells are not as harmful as most people thought, but rather the opposite. Now she will do some sharing for us about the senescent cells. Welcome to our broadcasting room, Dr. Nabora. Thank you so much for getting up so early for this live. So could you please make a brief self-introduction to the audience?
Yeah, thank you for having me. So I am a senior postdoc in Tian Ping's lab at UC San Francisco. I have been studying cellular senescence for the last 5 to 6 years during my training, and I'm wrapping up my training, and my next job hopefully is to have my own laboratory while I continue to study cellular senescence. And so we are a lab that studies the lung. So we focused on looking at the P16 or cellular senescent cells within the lung. However, I will say that we did find them with other tissues, but our studies were definitely focused on the lungs. And so what we do is we first generate the animals that actually can report and identify what we believe are cells that have characteristics of senescence. They express P16, which is a tumor suppressor or a cycling inhibitor. So this protein will stop cells from entering the cycling pool, so they won't proliferate. And so in the field of cellular senescence, P16 has been used as one of the markers, but it's not the only marker. There are senescent cells that also, express cyclin inhibitors, but there are other cyclin inhibitors, so it's not P16. So I just wanted to give you some sort of idea of the model and its limitations. So for example, in early development, senescent cells are thought to use cyclin inhibitor P15 instead of P16. And with our mouse model, we do not see any of these positive cells in early embryo. So that seems to go along with what's out there in the literature and has done by others. So our first approach, I don't know if you want me to continue or if you want me to answer the questions.
Well, actually, our share today is mainly for people without a background in biology. So maybe to them, it is more understandable to explain first at what are senescent cells, are they good or bad? It's
Yeah,
very simple
absolutely.
explanation is okay for them.
Yeah, so I'm glad that you shared the background of the audience. I totally forgot when I first logged in to ask, and that was one of the questions that I had. So senescent cells could be any type of cell. So you have your cells in the lung, the liver, the stomach. So as of now, we believe that any cell can change its current state and become a senescent cell. So it's like you can enter a different state. And what is the state? So when a cell has become senescent, it has decided to stop proliferating, to stop making more of itself. So cells have this ability to proliferate. If there is damage in the tissue and you need more epithelial cells, they can actually make more of themselves by dividing, right? So it's one cell becomes two, and then those become more, and then you make more in the tissue. So the senescent cell decides it doesn't want to do that, and it can't decide to not proliferate for many reasons. We believe that if there is DNA damage and there's too much of it, a senescent cell will stop proliferating or dividing because it doesn't want to contribute to bad damaged cells in the tissues, in the microenvironment. And so what we think is that throughout the lifetime of an organism, cells all over the body are deciding to become senescent cells. And then with age, you end up with a lot of these cells that don't proliferate, but they also don't die. They just sit around in the tissue. They are metabolically active, which that means is that they're consuming from the microenvironment and they're sending things out to the environment themselves. So until recently, we used to think that they were zombie cells, that they just lied around and did nothing. But we're learning that they're very active, and I believe that they're very chatty. They're communicating with other cells in the microenvironment. Yeah.
So in conclusion, that in common people eyes, the senescent cells, they do not divide, do not proliferate, they do not work properly, even they will secrete pro-inflammatory factors. But and in your findings, you find that they will communicate with other cells, and they also have some benefits too, right?
We believe they have benefits. And actually, other groups have shown that in the skin during wound healing, they can actually be helpful as well. They provide factors that support the growth of the skin fibroblast. So it's or the skin epithelial cell. So our finding is very unique because we found it in the lung. But the idea that senescent cells in young animals are helpful, it's an emerging idea. And so what we did is we injured the lungs of the animals, and we saw that the senescent cell can send out growth factors, so proteins that help cells grow and repopulate the airways of the lungs. Yeah.
And could we say that when we are young and healthy, these cells mainly play a positive role, but things change when we become much older?
Absolutely. That's the idea that I believe is most standing in the field, is that senescent cells tend to be very helpful when you're younger, but once they keep accumulating, and so why they accumulate, that's still it's still being investigated. We're not sure if the senescent cells are avoiding the immune system or in what mechanisms they have developed to stay alive and to not be cleared by the immune system. We know that they do in induced senescent cells in culture studies. They have shown that there's upregulation of some anti-apoptotic proteins. So what that means is that some cells could program cell death, and they could also hide from cell death or avert cell death by expressing certain proteins. And some senescent cells that have been induced in vitro actually express some of these proteins to avoid cell death. But that's still very under investigation. It's knowing why the cells accumulate in the tissue, how they're avoiding being cleared by the immune system, because the immune system's main job is to go around and survey the tissues within our bodies to try to clean up. If there's infected cells, they clean it up. If there's dead cells, they try to eat it up to make sure that our tissues are functional. But for the senescent cell, we're still trying to figure out how they're avoiding being cleared up by the microenvironment. How are they staying in the tissues if they're not proliferating? And we don't know 100% that they are contributing to the structure of the tissue in younger adults. Yeah.
Then I'm curious about what do you think are senescent cells and the aging of the body?
Yeah, so I believe that they are cells that have just been accumulating through life, but we don't know if the senescent cells can be induced later in life. That is something that I've been trying to answer. In my paper, we believe that maybe insults, like a lot of injury, you can cause more cells to enter the state of senescence. So that could be a possibility of why senescent cells are induced. But we would have to take old animals and do a lot of injury to see if we get more senescent cells in that age. But so far, the idea is that they're accumulating through age.
And then what do you think about the senolytics? Since that it is today's mainstream anti-aging therapy, since that our senescent cells are really beneficial for the younger people, then there are so many papers that are doing on the senolytics. Do you think?
Yeah, for now, I think I would stay on the side of caution for using senolytics, particularly in younger individuals, because a couple of reasons. The papers of published papers that have shown the senolytics can help aging consequences or improve longevity have done administration of systematic senolytics. So you're not just targeting specifically senescent cells within tissues, you're getting rid of majority of immune cells because a lot of the immune cells also express P16. And so when you get rid of immune cells in old tissues, you remove inflammation. So I think that is why the animals end up being a lot healthier, is because you're getting rid of a lot of the immune system. And the immune system is really bad for us, I believe, when you are older, because they're constantly contributing to inflammation. So not only do you have the senescent cell contributing to inflammation in the tissues, now you also have the immune system. So if you remove that, then I think the animals will do a lot better. So when you're young, we're not sure you want to get rid of your immune system. When you're young, you're not sure you want to, remove cells that probably are helping the tissue stay intact. So in our study, we use a senolytic, DNQ, dasatinib and quercetin, and we saw that the animals didn't regenerate their epithelial as well. So in young animals, at least in animals, we show that if you remove senescent cells, you can actually affect your regeneration program. So I would be a little bit more cautious in younger patients, but I think that my studies will show that we just need to continue to understand what a senescent cell is in vivo. A lot of the work that we've learned about senescent cells is during cell culture, and a lot of induced senescence with chemo agent drugs. So by damaging DNA of the cells, you can drive the cells to enter senescence, but that's not allowing for us to study the cells that become senescent in the body in vivo. And so I think that that's the disconnect. Maybe I will share that there is some companies in the US that have tried to use senolytics, and they've been failing at improving some of the aging consequences. And so we want to consider that and think about why would they be failing. And they actually didn't do a systemic administration of a senolytic. They did a local administration to try to kill senescent cells in the knee for osteoporosis, and then they end up not helping the symptoms. So then in considering this, I think it's because we don't quite understand senescent cells to the level where we should be trying to get rid of them right away. There's more to be learned from them.
Well, I have just to come up with a guess that the failing of these senolytics drugs that we have today, they might be caused by the aiming is not so prescribing, or does not have a how to say that?
I would say like affinity, right? It's not like there's an address and then you're sending it directly to the cell, right? Is that what you're trying to say, that the drugs might not be reaching the right cells?
Yeah.
Yeah, that's a big problem too. And that problem is because right now the field, to identify a senescent cell, we're using many markers, and then together we say this reality is that those markers could exist in other cells that are not senescent, like
Then
DNA
find
damage.
they didn't choose the right cells. Maybe they choose the wrong cells to kill so that they fail.
Right. We're still in the search for one marker that can define the senescent cell. And like I mentioned earlier, P16 is a very used marker, but it doesn't maybe missing some senescent cells that use other ways to pull themselves out of the proliferating pool of cells. So that's one of the reasons why I think it's very complicated to target these cells specifically, and why I think that it should be a collection of senescent cell studies of all different tissues, to really try to understand them so that we can target them better if we do. Because I think that if we can remove a cell that is constantly causing inflammation, especially in the context of aging, absolutely, we should do it. It's a good idea to try to remove them. I think senolytics in theory are excellent, but in practicality, it's challenging. And another reason why it's challenging is I believe the senescent cells are very plastic, in the sense that they can modify themselves in different environments. So if you want to target, let's say, inflammation in the skin and in the liver, just
They
a
will
wild,
try everything to prevent us to kill them. They're so clever.
they could, yeah, that's another thing. Or, the drug that's going to work for targeting senescent cells in the skin, it's different from the drug that's going to work to targeting senescent cells in the liver, because they're so plastic. There's different environments, so they're going to be different. But that's a really good point you bring out, that they can actually be very smart and escape. And so we're learning ways. There's just recent paper that came out of Japan that talked about an immune surveillance avoidance. So this senescent cells, they believe, use certain proteins to deactivate the immune cells so that they're not targeted, which is a really interesting mechanism, how these cells, maybe they're really smart and they're avoiding being cleared by the immune system, or maybe they avoid drugs. Yeah.
Difficult for us to use the same targets or the same markers to find all of the senescent cells. Things that first they're clever, they would avoid to being not killed, and but also they have different characteristics in different organs and tissues.
Yeah, and that's what we found as well. We didn't find like a black and white switch, for example. We found that their distribution of their characteristics was a range, and that adds complications to the studies. Yeah,
But the
but we did.
discovery of P16 is really a great one.
Yeah, and we did find that there is like a correlation with some of these, with the features of senescence. So the more P16 you express, the more senescent fixtures you have. So definitely that's there, we can't deny that. But again, it's not an absolute. That's what makes it more complicated and complex.
We're looking forward to new discoveries, the new P16s to emerge.
Yeah, well, we're hoping. What I'm working on now is trying to understand if these young P16 expressing cells are similar to old P16 expressing cells. So I want to know if they're having the same capacity to support regeneration process. That we don't know, and now we have the tools to study that. And so that's sort of where we're going next, trying to figure out how different are they? Can they help regeneration? And I think these studies are very key, because that would tell us that if one, they're different functionally, so killing them that would also tell us if it's a good idea to kill these senescent cells when you're young or not. And that's obviously a question your group has, and that's a question that many people in the field have, is how beneficial it is to get rid of them in young tissues.
Really a promising research destination, I thought, in the future. Well, our next question is about cancer. We know that the senescent cells can induce the immune system to kill cancer cells, but some anti-cancer drugs have the effect of removing senescent cells. Well, it seems a little bit paradoxical in this condition. I mean, the navitoclax or the dasatinib, they are the anti-cancer drugs, but now they also play another role as senescent cells clearer. So why will this condition happen?
Yeah, so this boils down ultimately to where the drugs are targeting, what processes in the cells. So these drugs tend to target what I was referring to earlier, this apoptosis pathways, so the cell death pathway. So they're trying to induce the cells to die, and they directly get rid of the proteins that protect the cell from dying. And this is why they work so nicely, because some senescent cells, not all of them, will upregulate what we call BCL2 proteins. And so these BCL proteins are, when the cell has a lot of them, the cell won't die. It's like an armor, a protection. And so Navi can come, the ABT series can come directly and break down that shield, so the cell ends up dying. But Navi, I believe, works, there's different types of them. There's like 263, 757. They work in different cell types, so they won't kill all of them. And there are some studies where you can see that within tumors, some cells become senescent because they're trying to avoid making more of themselves. So they remove themselves from the dividing tissue. But then in our lab, we're actually looking at cancer as well. You have senescent cells that actually can recruit things to the tumors and become supportive of tumor growth. So it's a double-edged sword. That seems to be what we call the pleiotropic nature of senescent cells, is that in one setting, it can contribute to tumor formation by recruiting to the area through the SASP, or the proteins that are coming out of the cell. And another way, it actually could help avoid cancer by themselves. The cancer cells typically, or the epithelial cells, can actually enter the state of senescence. I hope that was clear.
I quite agree with you on that the senescent cells are double-edged swords, as you just mentioned. Since that I heard some cancer patients who tried one therapy, which means to boosting or to rejuvenating the immune system to help clear the senescent cells, but they have developed other diseases like type 1 diabetes or autoimmune diseases. So these kind of condition, what do you think of that?
Yeah, I mean, that seems just really complicated. So if we think about the paper I described earlier, if you boost up, make a super immune system, maybe the senescent cell has mechanisms to suppress part of it and escape the clearance. But I would walk on the line of, I'm not an immunologist, but I would walk on the line of caution in trying to boost up the immune system in that way. I mean, we're learning through our COVID-2, COVID-19 patients that too much of an immune system could be really hurtful to cells. A lot of inflammation actually can be hurtful to tissues and render them inactive. In generating or boosting too much of the immune system, and you're teaching them to find antigens, you can make mistakes and teach them to find antigens that are actually self, and they'll recognize your own cells as bad, and that's why they target you, and then you end up with these autoimmune diseases. But I'm not an immunologist, so I'm not quite certain what type of training the immune system goes through to recognize your own cells as bad, and then you develop autoimmune disease. But I would imagine that these processes get disrupted if you end up supercharging the immune system, and then you end up attacking your own tissue. And autoimmune diseases come in many, many different flavors. There's very mild ones, and then there's very complicated ones. So if I were a patient, I would be a little bit more cautious of supercharging my immune system, unless I knew that it was very specific enhancement of the immune system. If I knew that like a vaccine, for example, that the immune system is going to get trained to find the cells with this one virus, right? Or this. So then for those reasons, I do vaccines. So I'm okay with using agents to amplify your immune system or supercharge the immune system. But it would be a little bit more cautious in other settings, like in just, I need to look more into these therapies because, again, I'm not an immunologist, so I'm not sure how they are amplifying the immune system.
Well, what I get from your attitude is that we need to be very cautious, and we could not use the drugs just like senolytics to clean our senescent cells unless we must do that.
Yeah, that's would be a good assessment. And I could be a little bit more conservative. And I think the more that I study the senescent cells, it is driving me to be a little bit more conservative, because it's sort of like when you become an adult, the more you learn, the more you realize I don't know a lot. And so I think as the field right now, we have learned a lot about the DNA, the chromatin structure of senescent cells. We learned about how their size changes. We know about how their organelles, the compartments inside of the cell change, but there's still so much more to be learned about their behavior, and particularly within the tissues. And I think that maybe in 10 years, hopefully we can have this conversation again, and it's in a nice brighter light where we can share that now we are moving forward and finding ways to target these senescent cells, and not necessarily, I think in the future, not to just remove them, but maybe to trick them to work for us. I think that they're so plastic that we might be able to use them to our advantage, because again, if you have so many in a tissue and you clear them all, now you left a tissue with missing a lot of cells. So it's sort of like you injure the tissue as well. So we have to consider that too. So if we can get these senescent cells to actually work in our favor and maybe stop them from secreting this damaging protein into the microenvironment, I think that would be a better option. But again, we're very much in the future here.
I suppose that I quite agree with you what you have just said, that I can understand the feeling that if I didn't know anything at all, I will think, well, it is simple. It's either black or white. But the more I understand about it, the more complexity that I found it will be.
Yeah, and It's kind of unfortunate. I think when people started looking at stem cells and, they thought, oh, that could be a really promising way to solve diseases, and now we're like many, many years into doing a lot of stem cell research, and we're finding that it's more complicated than what we thought. And that's typically how our natural world is and the biology. But that doesn't mean that we stay away from trying to study it. That means that we just need to continue our efforts in understanding these cells more thoroughly and see how we can actually take advantage of their existence, because it's very good tool to be able to have these sort of cells in tissues that can adapt to the needs of the microenvironment. So I think that we can use them to our advantage.
I quite agree with you on that. Um, what I'm also interested in is that you are specializing in long aging and regeneration. That except for the findings in senescent cells, what other discoveries or findings that you have achieved in recent years? Maybe we will focus less on that. So I would like to invite you to discuss more about other detailed discoveries on long aging and regeneration. For example, what are the differences or what are the similarities of the long aging with with the aging of other organs?
Yeah, so this is really interesting area for us, because in the I'll start out by saying what happens to the lung when it ages. So you have the upper area of the lung is where your airways are. And then when you get to what we call the distal part, you have the little sacs, the alveoli. And we have so many of them, a lot of real estate, surface area when we're younger. But when we're older, we tend to lose that. So we tend to have a lot less of these little sacs. And what that means is that you have less surface area to do the air exchange or the gas exchange. And so this is why when you're older, you tend to have more difficulties with breathing. A lot of the patients, when they're older, they end up having a lot of pulmonary diseases. So you're also more susceptible for some reason to lung diseases. So that's a big change that happens in the lung, because when it's young, the lung tends to regenerate pretty well. And you lose that capacity when you're old. And so our focus in our lab, our boss became really interested in learning about the communication that happens between the different cell types in the lung. So these alveolar sacs are made up of cells we call epithelial cells type 1 and type 2. And what we see is that with age, we're losing some of those cells. And the communication that we're interested is in the supporting. So those cells are in the alveolar sac, but there are other cells under those. They're called fibroblasts. And those are the cells that we're interested in. And we believe that those cells are contributing to the aging phenotype. And that was actually the project that I got started on when I got in the lab. But then when this senescence project was kicking off, putting that project aside. But there, what we end up doing is we used a premature model of aging with progeria. I'm not sure if you know the progeria syndrome,
Oh,
but you
please,
can.
but have some water now. We can wait for you.
Oh, thank you. Yeah, I don't know what is going on. Yeah, okay. Sorry about that. It's, I think, talking for too
Are
long.
off too, but as doing research is really tired for a young mom.
Yes. So we use this progeria animal. So you can probably, edit this a little bit, but my drinking water and coughing, I apologize.
So this is
We,
really okay for us. We're caring about our health. Health
yeah,
first thing that we need to focus on.
yeah, this is very true. So there are some people in the world that have a mutation and they aged very fast. So when they're 15, they look like they're 80. And it's incredible. Their skin, their cardiovascular system ages really quickly. And it's because of one mutation of a protein that holds the nucleus intact. I'm not going to go too much into the details, but we're able to take that. Scientists have taken that mutation and put it in an animal. So now we can make animal old. So usually in the laboratory, our animals are 3 years, they'll last, maybe almost 3 years. But here, 4 months, they're too old and they die. So we took the lungs of those animals and we believe that the fibroblast population, the supporting cells, if you make those old, they start to send out a lot of pro-inflammatory signals to the epithelial cells. And that's why the epithelial cell is not able to recapitulate the alveolar structure, and you end up with a lot less of these alveolar structures. So I did some very preliminary studies that has not been published. So I hope that now someone can go back and finish those studies to show that, because it's really important to determine how these supporting cells are helping the epithelial cells who do all the breathing for us, because how they're supporting them and how that support stops so that we can better regenerate, and not just in the context of aging, also like emphysema. So in emphysema, we also see the lung loses the alveolar structures, so you increase a lot of more air space. So you lose the cells that do all the breathing for you. So it'd be really important for many reasons to try to identify as how we can make the lung supporting cells help generate more epithelial cells in the context of aging and in disease as well.
Actually, I'm looking forward to the discovery of the further findings, since that I believe none of us will want to be the one who actually ate 16, but actually looks like 80.
Yeah, so there's unfortunately there is not a solution or a cure right now for these patients. People are working on it to try to understand how we can stop that, but it's really complicated to try to solve the problem. So it's, yeah, unfortunately.
Every solution or every drug in the history are actually have a tough way to go.
Yeah, absolutely. And then in our country, it's also like sometimes the lack of funding, the lack of people working in solving a problem. For example, for COVID-19, the whole world got together and tried to solve this problem. And we were able to now have vaccines. Your country has vaccines. We have vaccines. So we got together to solve this problem. But some of this other very rare diseases, we just don't have a lot of people like working on it and trying to figure out how we can solve that. Yeah.
An international collaboration might be.
Yeah, I mean, this global pandemic highlighted how important it is to do this collaborations, because, science is not individual thing. Is science doing good science, I think, takes community, and it takes many different minds and people, and what someone discovers here, others could use to discover something new. And so sharing ideas is something that drives me, and educating people, and I learn so much as well from others. So I think that having an international community, and this is one of the reasons why I decided to do your podcast, is to hopefully share something of what I know, and hopefully if someone's out there listening that wants to inspire them to be a scientist and help us in the field of cellular senescence, that would be great.
Spirit of science. Science don't have a nationality. This is what our Timepie will working on, to make the knowledge in the anti-aging area global, but as well as national.
Oh, that's wonderful.
Actually, listen to your sharing. Many of our followers also have some questions to convey. May I convey their questions to you?
Absolutely. Let's hear it.
And is, does long-term cellular energy deficiency accelerate aging? Or we come back to the topic of senescent cells.
So that was long-term what?
And
Sorry.
long-term cellular energy deficiency.
Yeah. Um, so they are. So, yeah, I mean, this is not my area of expertise, I'll say first, but from my understanding, they are actually deficiencies in mitochondria or the production that can give rise to different malignancies or different diseases. So being able to have a good reservoir of your energy, and the mitochondria is the organelle that gives the cells energy, can actually add to consequences of aging as well. So this is one of the things that goes array or dysfunctional when you in older cells. Another thing is the protein secreting pathways. You end up losing the ability for cells to properly send out and receive cargo. And this is why senescent cells are so interesting, because they are older, but they're still able to utilize these pathways really well, which is really interesting. And I think should be further investigated. You also have, like, I will add this is not in the context of aging, but in the context of cancers, cells end up utilizing different ways of producing energy for themselves. So metabolism changes in cancer cells. So metabolism and how cells make energy is very important in driving different pathologies, and definitely in the context of aging. We have two different modes of producing energy. There's the very oxidative phosphorylation that utilizes oxygen, and then the glycolytic one that doesn't. And we know that some cancer cells prefer one pathway over the other. And there's big efforts in using that to try to kill cancer cells, is by starving them, by blocking the pathways that they use to produce energy. So one can think of, in the context of aging, we can try to enhance pathways of energy production in the cells so that the cell could be more rejuvenated in a sense. Yeah.
Just mention about the mechanism of the similarities between cancer cells and aging cells. But what could our ordinary people get? Can we get some useful information from the cancer cells to help us prevent aging?
Yeah, I think so. So the cancer cell, I think of it as an immortal cell. So it lives forever, right? So figuring out how the cell, if you keep giving it nutrients and support, this cancer cell just won't die. So I think there's a lot to be learned on how they're doing that. How are these cells not getting tired? How is their energy production mechanisms not wearing out? And I think one way we're learning is that cancer cells can actually utilize different pathways of energy production. So maybe in older tissues, we need to enhance those pathways. But then again, we have to walk on the line of caution, because we don't want to give older people who already are susceptible to cancers more cancer, right? So it has to be a nice balance. Yeah.
Well, the next question that our audience is very curious about is that quite personal. What do you do in your life to improve your health, to activate your rejuvenation, or reduce the accumulation of senescent cells? Could you give us some suggestions?
Yeah. So I will say that, I look a little older than I am because I have kids, but I do try to be healthy. Um, in our country, we have, I think, very weak laws in protecting the food that we consume. So I think there's a lot of chemicals in our food. So one of the things that I do is I'm very active in selecting carefully the food that I eat. I want to make sure that there's no harsh chemicals used to grow my food. So I think what I consume is one of the ways that I try to stay myself really healthy. My family and I, we do a lot of hiking. I drink a lot
Hiking.
of water. Hiking, yes. Even my little tiny one, I put her on here. We go to the mountains, and I do a lot of hiking to try to stay healthy. In terms of what I consume, water, the last few months has been really difficult to do. More of I used to walk 5 miles a day, but now after the
Five
baby,
miles a day
yeah,
with a baby or two.
well, yeah, now it's more hard because now the baby is smaller, but later I can push the stroller with the baby. So I try to be more active because I think that activity helps the cell. I think that good nutrition will help your cells stay healthier longer, and avoiding compounds. So we know, like, these cancer drugs are damaging the DNA or damaging part of the pathway and to kill the cells. So some of the things that are in our environment can also kill our cells. Like in California, there's a law that when you buy clothes, they have to put fire retardants. So there's chemicals all over our world, and so that we don't fully understand. So it's almost impossible to avoid them. But when I become knowledgeable about them, I try to avoid them. And so that's the very little that I can do to try to protect myself and my family.
So there is no supplements.
No, I try to supplement as much in my food. Um, but I do drink a lot, like tea and coffee. So maybe it's not a good thing, but maybe it is. I don't know. But I I don't have, unfortunately, to share a secret on, like, I drink these tablets and they make me better. Not at the moment. I do know people that believe they drink these things that make them younger. Maybe it's true, but I just don't do that myself. Maybe your audience members, if they know something, they can share with me.
I will. The next question has come back to the academic one. Whether the changes in the microenvironment in aging long can be used for mesenchymal stem cell therapy.
Yeah, I think that we need to learn more about thoroughly what those changes are, because there's quite a bit happening. So we're not tracking the changes in a longitudinal way. What we'd end up doing is looking at the consequence. We already looked at what happened. So we get along and we see, okay, we have more airspace, we have less of these cells. But what we need to learn first is, how is this mesenchymal population contributing to aging during the time? And once we learn how it's contributing to aging, is it whether it's sending out too many signals for so much proliferation in the epithelial cells that they became exhausted, or if they're not sending out in the microenvironment the collagens, the laminins, things that are supposed to have the structure of the microenvironment properly. We don't know that. Or are they sending signals to recruit too many immune cells? There's just so much, I believe, that is still to be learned about how these cells contribute to aging. And once we learned about that, then we can think about how to help them better support the aging lung. Very good question. And that was a lines of what I was trying to study. And so I just now that we have this aging ink bright animal that we generated, we're starting to do a lot of we do co-culture experiments in ex vivo outside of the body in a matrix. We put the mesenchymal cell and then we put the epithelial cell together, and we're trying to figure out which population is more supportive or less supportive. And that's the beginning. So years to come, hopefully we can have a little bit more of idea how the relationship between these supporting cells of the lung and the actual epithelial cells who do the job of the lung, right? We want to make sure that in old tissues, we can get more of these type 1 or type 2 epithelial cells because they're the ones that are going to do the breathing for us. And we want to continue to be able to breathe properly. Yeah, very good question.
As you just mentioned that you used co-culture or not co-culture. Our audiences, they're really careful about any detail in your paper or in your lecture. They are wondering that why do you use basal cell rather than secretory cell co-culture in the research?
We use the SCGB1A marker of the ciliated cell. So why did we use that cell line? Those are the ones. So for our study, we were focused on the upper airway. So we wanted to use the progenitor cell that will repopulate the airway after injury. But now I'm doing studies where I'm looking at the distal part of the lung, where I'm using SPC positive cells. So I'm trying to see if I can re-injure the alveolar regions and how P16 positive cells, will they support regeneration for those type 2 cells? Yes. So I chose that one specifically because I wanted they are the ones that repopulate the lung, as is my understanding. They will make, so the basal cell will make the other cell types. And when I do my injury, I delete everything and the stem cell is left behind, and then they will make more of all of the other airway cells. I hope that was clear.
Really clear. Although not willing to, our interview comes to an end. It's just really appreciated that you accept our interview and share with us detailed and fruitful interpretation on citizen cells and the other drugs or cancers or other your research results on lung regeneration. I hope that we could get close contact in the future and wish everything goes well with you and your baby.
Oh, thank you so much. And I appreciate the invitation. I hope that I was able to share some good information, and I apologize for being so sleepy. It's getting close to evening time, and this was a great opportunity. I think that I'm really grateful that you reached out, and hopefully there are young people in your audience listening and get encouraged to enter the field of cellular senescence.
Thank you so much.